This is a final run through of all the topics for the 2022 higher tier of AQA GCSE Combined Science Biology Paper 2. In other words, I'm missing out anything in the not assessed section of the advanced information, and the major topics are on green slides. So anything that's on a lilac slide hasn't been ruled out, it can still come up, but usually only as a low tariff question worth a couple of marks. I'll say at the outset that this is a mix of some new recording and also some bits I already had recorded, so I'm sorry in advance if it's really annoying that the audio quality is going to change occasionally. Before we get started on the biology, a couple of quick reminders. Firstly, it's important that you're writing in black pen, so if you don't have one, make sure that you go and ask someone before you get into the exam. Your paper is going to be scanned and blue pen just does not scan as well. For similar reasons, it's really important that you stay within the box on the exam paper because otherwise your work might not be read by the examiner and they might not be able to give you credit for it. In any calculations, watch out for sneaky units that could be worth an extra mark and trip you up. And remember that for any extended response question, it's absolutely encouraged to answer in bullet points. There aren't any marks for full sentences, and it's just going to make your work clearer and easier to mark. If you see the command word evaluate, then you need to give both sides of the story and sort of compare them. Try to add value to whatever is in the table. So for instance, saying, oh, this number is 5 times higher, not just saying, oh, it's a big number. And make sure that you write a conclusion because that conclusion is always worth 1 or 2 marks. If you have to write a method, say for one of the required practicals, then make sure that it logically flows and that it actually makes sense and would allow someone to get the data that they're trying to collect. You are going to be assessed on both working scientifically skills and mathematical skills, so make sure that you've revised those this evening as well as the biology content. You should be able to identify any variables for the practicals that you're doing, so the independent variable being the thing that you change, the dependent variable being the thing that you are measuring or observing, and the control variables being the things that you keep exactly the same every time to make it a fair test so that you get valid data. If you repeat the experiment using the same method and you see the same pattern, then that's an example of repeatable data, whereas reproducible data might be produced by a different group of scientists, or they might be using a slightly different method to the method that you used. Resolution is the smallest difference in data that you can detect using your equipment, and we like using computerized methods of data collection because they are more accurate, rapid, and sensitive. If you have to turn data from a table into a graph, then make sure that you are plotting all of the data, even if one of the data points is at the origin or 0, 0. And remember that your line of best fit absolutely can be curved. So look at the shape of the data. If it's in a straight line, then draw a straight line. But if it's not in a straight line, then draw a freehand curve instead. When you collect that data, there will always be some error around the true value. So this could be a systematic error where there's an issue with your equipment and maybe say you have a balance that's always measuring half a kilo lighter, in which case you would just need to add half a kilo to all of your measurements. Or you could have a random error. So this is where we have a scattering of data around the true value. And so the way that you get as accurate as possible is to make multiple different readings and then calculate a mean using these. Following the biology exams in particular, social media is always filled with people being absolutely outraged that they've been asked to do maths in a biology exam. So don't forget, 10% of the marks are going to be for maths at an appropriate level. So just adding things up or multiplying them together doesn't count. We're talking things like giving your answer as a decimal or giving it in standard form or answering in the form of a percentage, making sure that you round to the correct number of significant figures. Just make sure that you've had a little look at this list beforehand so that there's nothing on there that's going to come up that's going to just trip you up because you didn't know that you might be assessed on it. If you have the opportunity to have a look at your specification this evening, then the full list of requirements is in chapter 9 at the back. The first topic in paper 2 of AQA GCSE Biology or Combined Science is the Homeostasis and Response topic. Homeostasis is the name we give to the regulation of the conditions inside a cell or a whole organism in order to maintain the optimum or best conditions for function in response to changes both internally and externally. What this means is that the environment is changing constantly, and for a cell to be able to work at its absolute best, it must be able to overcome those changes. Many of these changes are controlled by negative feedback loops. Negative feedback works when a change in the environment is sensed and a response is created. For instance, maybe a hormone is released. That hormone will be released until the point where that change in the environment has been overcome, and then it will stop being released. The 3 named examples of changes you need to be aware of for GCSE biology are blood glucose concentration, body temperature, and water levels. Homeostasis pathways have three key parts. Firstly, specialised cells called receptors detect a stimulus, which is a change in the environment. They pass this information to a coordination centre like the brain, spinal cord or pancreas, which is able to decide what to do next. The response is then carried out by an effector, such as a muscle which may contract or a gland which may release a hormone or enzyme. Homeostatic responses may be either nervous responses brought about by the nervous system or chemical responses brought about by the endocrine system, or a combination of the two put together. The endocrine system works by releasing chemical messengers called hormones into the bloodstream, and the changes it brings about happen slowly, but they tend to last for a long time. In contrast to this, the nervous system relays information along neurons using electrical impulses. It's much more rapid, but the responses it brings about last for a shorter amount of time, so it's good for an emergency response which doesn't need to carry on happening. The nervous system has been taken out of the combined science higher tier papers, so although you need to know a little bit of detail in terms of how it compares to the endocrine system, you're not going to need to know things like how a reflex reaction works. But your first major topic is the endocrine system. So this is the hormone system. So we know that endocrine glands secrete these little protein packages called hormones, and these are chemical messengers that are carried in the blood until they reach a target organ. There are 6 specific endocrine glands that you need to know the locations of. In the brain, just behind the bridge of the nose, is the pituitary gland. Then your pancreas is part of your digestive system. It's the small leaf-shaped organ that also produces a number of the digestive enzymes. Your thyroid is in the neck by the Adam's apple, and your adrenal glands are sat on top of the kidneys. And then depending on your sex, you have either ovaries or testes. The pituitary gland is called the master gland because it's a gland, but it acts on other glands. So the pituitary gland releases some hormones, and they move through the blood to the other glands and act on them, and then those other glands produce further hormones, so we can have a kind of cascade effect. Your thyroid gland produces a hormone called thyroxine, and this is responsible for your basal metabolic rate, that underlying metabolic rate that you have all the time, regardless of how much exercise you're doing or what you're eating, and it's really important for growth and development. So people that have an underactive thyroid gland often have difficulties with their growth. Thyroxine is controlled by a negative feedback loop. So basically what this means is that the thyroid gland will produce it and then monitor until there's too much in the blood, and at that point it will stop producing it and wait for the levels to come back down. Your adrenal glands make adrenaline, which is important for times of fear or stress because it manages that fight or flight response. It increases the heart rate, and this is important because it means that your muscles and brain can get more oxygen and more glucose to do more respiration so that they have more energy. It's really important that your blood glucose levels remain more or less constant, both because you need a regular supply of energy and also because if they fluctuate too much, it can cause damage to your cells. Your pancreas is responsible for both monitoring and controlling the level of sugar in the blood, and it does this by using 2 hormones called insulin and glucagon. When the blood glucose level rises too high, the pancreas makes insulin, and this causes the permeability of your cells to increase so that sugar moves into the cells, particularly in the liver and the muscles. And here that excess glucose is converted into a large storage polymer called glycogen. If you're taking the higher tier, then you also need to know about glucagon, which is the hormone that works in reverse. So when blood glucose levels get too low, the pancreas makes glucagon and glycogen is turned back into glucose. We can see this in action here. Blood glucose level rises, so the pancreas is stimulated and it makes insulin, which it secretes into the bloodstream. This causes glucose to be taken up from the blood and stored in the liver and muscles so that blood glucose level falls. Then, if you're taking higher tier, you need to know that the pancreas will be stimulated again, but this time it produces a different hormone. It makes glucagon, and this causes the liver to release glucose, so the blood glucose level goes back up again. Linked to blood glucose levels are type 1 and type 2 diabetes. Type 1 diabetes is often diagnosed in much younger patients because it isn't caused by a lifestyle risk factor. It has a genetic component and it often runs in families. If you have type 1 diabetes, then your pancreas stops making enough insulin, and this means your blood glucose levels can go scarily high. In order to be treated, you have to take injections of insulin, and without these, you could even die. It's diagnosed with the 4 T's. Because your blood sugar levels get so high, the only way to get rid of that blood sugar is to urinate it out, so you need the toilet constantly. And also because you're urinating so much, you need to drink constantly, so you're thirsty all the time. Because so much of the sugar that should be going into your muscles as glycogen is being wasted and urinated out, you're tired all the time because you don't have enough energy, and you often lose weight as well. In type 2 diabetes, the pancreas is still producing enough insulin, but because the insulin levels have been so high for so long, the body cells stop responding. Type 2 diabetes is often associated with obesity, which we can call a risk factor, and in order to treat it, we use a carbohydrate-controlled diet and an exercise regime. It's really important that the level of water in the blood is controlled, because if it isn't, then the red blood cells could either shrivel up or burst. This process is called osmoregulation. We naturally take in water when we eat and drink, but then we lose some in an uncontrolled way by breathing out the waste products of respiration and by sweating, and we lose some more in a much more controlled way from our kidneys. In order for our bodies to remain healthy, the amount of water that we take in and lose must be balanced each day. During puberty, the levels of reproductive hormones like estrogen and testosterone begin to rise, and this causes the development of secondary sexual characteristics like breast growth and facial hair growth. In females, the main reproductive hormone is estrogen, which is produced in the ovaries, and in males it's the testosterone, which is produced in the testes and stimulates sperm production. The menstrual cycle is controlled by 4 different reproductive hormones. Firstly, follicle-stimulating hormone, or FSH, is produced by the pituitary gland in the brain, and this causes an egg in the ovary to mature. Then, luteinizing hormone, or LH, stimulates ovulation where the egg is actually released. LH is also produced by the pituitary gland. Estrogen and progesterone are both involved in maintaining the uterus lining, and these are produced in the ovaries. At the start of the menstrual cycle, as progesterone levels fall, the lining of the uterus is shed. Follicle-stimulating hormone causes an egg to mature and also stimulates the production of estrogen. This causes the uterus lining to regrow, ready for a fertilised egg to implant. Estrogen inhibits FSH and it promotes LH, and when LH is produced, this leads to ovulation. High progesterone levels maintain the uterus lining, and if pregnancy occurs, progesterone levels will remain high so that the uterus lining remains intact. If you're taking higher tier, you also need to be able to describe the use of sex hormones in fertility treatments. Fertility drugs contain follicle-stimulating hormone and luteinizing hormone, which together stimulate egg maturation and ovulation. Some women may be able to conceive naturally after receiving a dose of fertility drug. If this is not successful, then the woman may undergo in vitro fertilisation. She takes the same fertility drug, and then the eggs are harvested, and they're fertilised in a laboratory instead. After they develop into embryos, several embryos may then be implanted back into her uterus. Although fertility treatment does give women with fertility problems an opportunity to have a baby, it's stressful both emotionally and physically, and the success rates are low. As a result of this, we tend to implant multiple embryos at the same time, but this means that if multiple embryos implant successfully, it can lead to multiple births, and having twins or triplets is much more risky than a single birth. Within the reproduction section, AQA have taken out section 1, 3, 4, 5 and 6, but they haven't taken out section 2, which is meiosis. So although this isn't a major topic, I think it's fairly safe to say that we are expecting a low tariff question which directly asks you about meiosis. You need to know about 2 types of cell division. For paper 1, we talked about mitosis, which happens in all body cells and allows them to make more body cells for growth or for repair. It starts with DNA doubling, and then the cell divides once, so there are 2 diploid cells made. These cells are identical to each other and identical to the cell they were made from. Meiosis we only talk about in paper 2, and it happens in the reproductive organs, so the testicles and the ovaries. It makes sex cells, also called gametes, so that's pollen, egg cells and sperm cells, depending on whether we're talking about plants or animals. The DNA also doubles at the start, just like it does in mitosis, but then the cells divide twice. So rather than producing diploid cells with the same amount of DNA as the parent cell, it produces haploid cells, and 4 of these. The cells are different to each other and also different to the original cell because they have half the amount of DNA that the cell started with. Classification is the science of putting species into groups based on their shared characteristics, and you need to know about 2 systems, the old school taxonomic hierarchy system and also the 3-domain system as proposed by Woese in 1990. In the 3-domain system we have eukaryotes, including animals, plants and fungi, then the true bacteria, and finally the archaea, which truthfully are as different from true bacteria as they are from us, but according to AQA they're primitive bacteria usually found in extreme environments. We call these types of archaea extremophiles, literally things that like extreme environments like high temperature or high levels of salt. In the traditional taxonomic hierarchy system, we started out with 5 kingdoms: animals, plants, fungi, bacteria, and Protoctista, which includes slime moulds and Plasmodium, which causes malaria. Each kingdom is then split into a number of phyla. So for instance, for humans, our phylum is called the chordates, and this involves anything that has a spine and a spinal cord. Phyla are then split into classes, classes into orders, orders into families, and then families into genera and species. The last two of these categories are used to create a species binomial name. Now, you might have heard this called a Latin name before, because a lot of binomial names are in Latin, but lots of them aren't, and we should really use the proper name for them. So this is made out of the genus and the species, and the genus has a capital, the species doesn't, and there's a space between them. So the binomial name for humans is Homo sapiens, and this system was developed by Carl Linnaeus. The final unit in Paper 2 of Biology is Unit 7, Ecology. An ecosystem is the interaction of a community of living organisms, so the biotic factors, with the non-living or abiotic parts of their environment. In order to survive and reproduce, organisms require a supply of materials from their surroundings and from the other living organisms that are there. Animals and plants may compete intraspecifically, which means within their own species, or interspecifically with other species for both biotic and abiotic factors. Where a species is able to survive and thrive will be a combination of both the biotic and the abiotic factors. For instance, an animal species may not be able to survive in an area where there's a superior competitor taking all the food, but then also where it lives could be controlled by something like the temperature being too hot. Or as a plant, you might not be able to survive in an area that doesn't have sufficient light, so you can't do photosynthesis, or where there aren't enough mineral ions in the soil. When we talk about interdependence, we mean the way that different species interact with each other in order to be able to survive. So for instance, insect A might be a pollinator like a bee that drinks the nectar of plant C while it's pollinating it, and then mammal B might eat the fruit. And plant C needs both of those animals because even though they're feeding on it, without them it wouldn't be able to make fruit because it wouldn't be pollinated and it wouldn't be able to spread its seeds. A stable community is one where all of the species and environmental factors are in balance so that population sizes remain fairly constant. Feeding relationships in an ecosystem can be represented by food chains, which can be put together to make a food web like this one. In each food chain, the direction of energy transfer is shown by the arrows. Every food chain begins with an organism that can synthesize molecules containing energy, and we call these organisms producers. Most producers are green plants or algae making glucose by photosynthesis. Producers are then eaten by primary consumers, which we can also call herbivores, and then primary consumers are eaten by secondary consumers, and secondary consumers by tertiary consumers. We also can say that anything that kills and eats other animals is a predator, and anything being killed and eaten is a prey. In a stable community, the numbers of predators and prey rise and fall in cycles. The ecology required practical centres around measuring the population size of a common species such as clover, usually in response to an abiotic factor like light, moisture content or pH. To estimate the population size, you use this piece of equipment which is called a quadrat. It's a square frame and it often has divisions inside to help you to estimate what percentage of the quadrat contains the plant you're sampling. Depending on your exact investigation, you may be comparing two separate locations or looking at a transect, which is a long straight line where an abiotic factor changes gradually. If you're comparing two locations, you use random sampling at each location to ensure you have a fair representation. Split the area into a grid using coordinates and then use a random number generator to tell you which squares to sample. For instance, square B1 and C5. At each location, you sample the coverage of the plant using the quadrat, and you also take a reading of the abiotic factor, such as a light reading with a light meter or measuring acidity with a pH probe. If you're looking at a factor that gradually changes, such as moisture content as you get closer to a river, then instead you use systematic sampling. You use a long tape measure to make a line called a transect, and then you put down the quadrat at regular intervals along the way, say every 5 metres. Again, at each location, you survey percentage cover and your abiotic factor. There are some opportunities in here to test your math skills, and these usually involve working out the area, either of the whole sample site or the quadrats that have been surveyed. These are usually quite easy calculations to do, but also easy to make silly mistakes with, so do check your working. Materials are cycled through ecosystems as part of both biotic and abiotic processes. You could be asked about the water cycle, which provides fresh water for animals and plants, or the carbon cycle, which contains biotic and abiotic processes. The total amount of carbon in the atmosphere, in the form of carbon dioxide, is increased by respiration from animals, plants, and microorganisms as they break down glucose and release carbon dioxide.
It's also released by decomposition, in which microorganisms break down dead plants and animals and then release carbon dioxide into the atmosphere as they respire and mineral ions like nitrates into the soil. Carbon dioxide levels are also increased abiotically by the combustion of fossil fuels. Meanwhile, carbon is removed from the atmosphere by photosynthesis, in which green plants and algae absorb carbon dioxide and make glucose. They then store this as starch or use it to make fats, proteins and cellulose. Carbon dioxide is also removed when it dissolves in oceans and forms into carbonates, which are locked up in sedimentary rocks, and also by the production of fossil fuels and peat. Carbon can also be transferred between plants and animals when animals eat plants. Biodiversity is all about having the maximum number of species possible in an ecosystem, and the reason that we care is that this makes the ecosystem much more resilient and able to resist change. For instance, if a disease causes a particular plant to have reduced numbers, and it's the only food source in an ecosystem, then many herbivores may die, and this has a knock-on impact up the food chain. However, if there are several other plant species, then the herbivores may be able to survive for a year by eating those instead, while the population of the first plant recovers.
There are lots of things that humans can do to protect and preserve and encourage biodiversity, including running breeding programs for endangered species, protecting and regenerating rare habitats, and also reintroducing field margins. So in other words, rather than having one huge farm where you just have a massive field and no gaps in between, actually having little hedgerows around each farm so that things can be living in those hedgerows. Unfortunately, there's also plenty that we as humans do to make the biodiversity situation worse, including polluting the water with sewage, fertiliser and toxic chemicals, which could either be responsible for killing animals directly or for causing eutrophication to happen so that the oxygen levels in the water go down. We can pollute the air from smoke and acidic gases, and also we can pollute the land from landfill and from toxic chemicals.
Carbon dioxide, methane and water vapour are all examples of greenhouse gases, which are molecules that will trap infrared radiation, causing Earth to warm up. Having a little bit of them is a good thing because it keeps our temperatures stable and also high enough that we can actually live on Earth, but unfortunately we now have too much of them, and this is leading to global warming. These greenhouse gases form a blanket around the Earth, and it's possible for small wave radiation like visible light, UV, gamma and X-rays to get through this, but when that radiation reaches Earth, the Earth absorbs it and then re-emits it as a longer wavelength wave, like infrared radiation. Now that infrared radiation is too large to easily pass back through the blanket, and although some of it does get through, some of it is trapped and warms up the atmosphere and Earth's surface. The rising temperatures caused by this are leading to lots of serious consequences. As the ice caps melt, sea levels rise, and this can lead to flooding and other extreme weather events like droughts and hurricanes. Polar habitats are being lost, and with them many species are going extinct.
That's it. Thank you very much for watching, and I hope that you're now feeling more prepared for tomorrow's exam. Good luck with any last minute revision that you're doing this evening, and I hope that you manage to get a really good night's sleep and go in there tomorrow and do your very best.